Hello, it's my pleasure to introduce Professor Tim Behrens for this keynote lecture. Tim is from the Oxford Center for Functional MRI of the Brain. He's the Deputy Director of the FMRIB. He's a Professor of Computational Neuroscience at the University of Oxford. And if you've seen Tim around, you may wonder why he looks so young. And that's because he is. If you haven't, but you know his work, you might be surprised to find that he's under 76 years old, which is his age index. But I'm not going to review all those 76 papers for you. I hope Tim will talk about just a few of them, at least. But I just wanted to point out a couple of the areas in which I've been particularly impressed with his work. Tim's done some fundamental work in diffusion imaging, both in developing the methods, starting probably in 2003 or earlier, which has led into a very productive area of work in anatomical divisions and brain connectivity, and using those methods to understand the architecture of the brain. He also has made substantial contributions to architectures of decision-making, work on counterfactuals, work on hierarchical models of learning and changing environments, work on social decision-making from a rigorous computational perspective. So he's continually evolving and really surprising all of us, I think, by moving through these areas with grace, and also having a lot of fun along the way. I asked him just now what his favorite paper is, and he says it is what is the most interesting part of the brain, which I also picked out as his most fun paper, which might tell you a lot about Tim's personality. And maybe you can ask him in the Q&A, what is the most interesting part of the brain? I don't know. He's received grants from the MRC, from McDonnell, EPSRC, and as a Wellcome Trust Senior Fellow, a senior editor of eLife, and an editor at PLOS Biology and PLOS Computational Biology. The title of his talk today is Storing, Using, and Updating Knowledge for Behavioral Control. So please join me in welcoming Dr. Tim Behrens.
Thank you. Very exciting and nerve-wracking to be here. Where's my controller? So I'm just going to start the day by introducing you to the cast of characters who are you're going to see the work of today. And you'll see them again as we progress through and I show you their work. But I thought it was important that I showed you their faces beforehand because in case it might affect the way you evaluate their work. Because 3 of these people are immigrants to the UK. And you might notice just by looking at this slide that the other 2 are ginger, and so you should treat anything I say with a great deal of skepticism for the next 40 minutes or so. So these are Helen, Alexandra, Jill, Mona, and Erie, and I'll show you them later on as well. We're interested, and have been for a while, in this general problem, in the idea of how you make decisions in difficult worlds, how you plan through these worlds, how you learn about these worlds to optimize your behavior. It's, as you all have seen in all of behavioral neuroscience, it's an important constraint on evolution and on evolutionary success. And recently, I've been interested in the neural codes that might support storing these models, and in particular, how do I go about representing these links, the relationships between different parts, which things lead to what, and which things are related to what in the world. And that's what I'm going to talk to you about today. It's an important problem, I think, in many parts of cognition. It's obviously important in simple reward learning. That's the basis of associative learning. It's important in social behavior if you want to know where you are in your social hierarchy or your social network, so you can decide who to approach about what. It's obviously important in executive planning. If you want to know how to get somewhere, you need to know how things are joined up. And I think that some of the codes that I'll be talking about at the end of the day today are important for really basic things like understanding this talk, for understanding the relationship between different concepts in your world. Okay, so if you're going to, let's start at the beginning with this very simple association and ask what kinds of neural codes might we be interested in if we're looking at something that is representing an association between two objects in your brain? And this association is between Starbucks and coffee. It's probably an association that you've used just today. And you might think that when you first formed that association between that Starbucks sign and that coffee, you did so by taking some cells that responded to that green Starbucks sign and some cells that knew what coffee tasted like and making a connection between them with some excitatory synapses, so that when you see that green Starbucks sign, it will activate the pool of neurons for coffee, and you will know that Starbucks leads to coffee. I'm going to call that an implicit way of storing an association. It takes two neural codes that already exist and forms a connection between them. There's another way you might think of doing it. You might think of forming a new pool of neurons that themselves will encode that association. And so you may have a new pool of neurons that says these two things are associated, and then it then activates one when it sees another. I'm going to call that an explicit code for an association. So let's go back to these implicit ones and notice that it's not an easy thing to do as you choose to have more and more relationships. So when I see a Starbucks sign, I might be interested in a cup of coffee, but I might also be interested in this nice piece of red velvet cake. And if I then try and build the same kind of algorithm for associating these things together, then I'm going to end up with a little network like this. But unfortunately, when I start activating the Starbucks sign, it will activate the coffee and the red velvet cake, and you'll end up with an orange blur of activity that is very hard to disentangle. You won't know which population is representing what. So that doesn't seem like a good idea to go that route. But there are perhaps ways that you can save this route, and those saving ways are what I'm going to talk about for the first few minutes of my talk. And then this, if we think then again about the same problem in the explicit case, you could choose to use what I might call an explicit local code, where you have different cellular populations that encode each link. And so these cells are local in that they encode just one link. Or you could, if you wanted to, choose a more distributed global code where one cell population encodes something global about the structure of the associations, or encodes parts of multiple different links. I'm going to call that an explicit global code, and that's what I'm going to talk most about in the second half of my talk. Okay, so I'm going to start with controlling local implicit relational codes, those first types of codes in the sensory cortices. And this is work done by Helen Barron, who was a PhD student, is a postdoc now, and she's done work in human fMRI that you're about to see, and is currently doing the same thing with rodent optogenetics. So this is here are those two pools of neurons again, and I'm going to try and make a way of assessing whether this link exists using an fMRI scanner. So here's this. So here are these two pools of neurons, but instead of them being Starbucks and coffee, I'm going to go for a rather more controlled experiment with different shapes, and I'm going to pair shape A with shape B, and I'm going to pair shape C with shape D using basic behavioural conditioning in my subjects, and so my subjects will know those pairings. And there's one other thing to note, which is that those shapes are rotationally invariant, which means they can be presented to the subjects within any orientation. So obviously, we think that if that's the case, a new excitatory connection forms, then there'll be some overlap now in the representation of A and B, because when you excite A, it will also provide excitation in the B cells, which means we might use fMRI techniques for measuring representational overlap to look for this link. And obviously, I could use RSA approaches or multivariate pattern approaches. But we've chosen not to do that for various reasons that I can go into later. Instead, we're going to use a repetition suppression approach. And so this is the type of measure I'm going to be using for the first bit of this talk. I'm going to very rapidly present two of my symbols, and they might be a paired symbol, two paired shapes, BA, or they might be two unpaired shapes, CA. And I'm going to say if there's a link in this brain region, then the pair BA are going to have lead to suppression, to less signal than the pair CA, and so my measure is this cross-stimulus suppression is going to be the difference between the two things. And that's going to be an index of whether there's an association between these two things in the brain. And indeed, there is. So here in the region of the brain just anterior to the lateral occipital complex, in a region of cortex that knows about rotational invariance, there is a suppression between the two paired shapes, suggesting that there is some pairing there in that cortical network. So that works in this brain region, and it also works if I choose rather simpler stimuli that are not rotationally invariant but have color. It also works in early visual cortex, and it also works in a large number of other brain regions that we've shown in these two papers here. So somehow it can be a general mechanism for indexing associations across cortex. But as I said, this is a really terrible strategy. It's not a good thing to do. It's because if you start making lots of different pairs, the activity will start flooding into each other, and we'll start losing any precision for what the neurons are coding. So there's been various suggestions about what you might do about that, but one very attractive one is that you might build anti-associations or anti-memories into the network, such that the memory is stored in the network in the excitatory connection, but the activity is prevented from flooding around by a mirrored inhibitory connection. This sounds like a very attractive idea. It allows you to store lots and lots of memories or associations in a network. There are some interesting computational analyses that can show that. But is there any evidence that it might be true at a synaptic level? And I think that there is. So if you go and record from one of these cells, you could make some recordings inside the cell using patch clamping, and you can record separately the excitatory and inhibitory currents. And if you did so, you'd find out they were balanced in a very precise way at a millisecond by millisecond resolution, as if the same signals were coming in both on excitatory and on inhibitory synapses. It's also the case that if you go and disrupt this balance, I'm not going to explain this graph, but what it shows is if you then disrupt this balance between excitation and inhibition and leave the network for several hours, then automatic processes in cortex will re-establish that balance within 3 hours. And lastly, if you take some very clever theoretical approaches to modeling these data, you can show that all of these effects can be accounted for by plasticity at inhibitory synapses, which learn to match exactly the plasticity at excitatory synapses that have stored whatever information it is you're storing. And so the question is, can we measure something as detailed as this, some synaptic level code as detailed as this, using human fMRI? And it's a very daunting thing to suggest you might be able to do, and I'll be interested to see who believes we can. So the approach that we've taken to doing this kind of thing is to start off with to leave the subject with the association for several hours here overnight, and measure it again and show that the association has gone in cortex, but is still present in behaviour, suggesting that something has changed in that network in those hours. And so obviously that could just be that the whole thing has moved to hippocampus or some other part of the brain. So the question is, can you get that memory back in cortex with any kind of manipulation? And the obvious one that you might try if you were a rodent if you were a rodent neurochemist is to introduce some kind of blocker which would block GABA, and that would allow these populations to start overlapping again, and so you could start to measure the overlap in representations. And we don't have those GABA blockers easily available in humans, but there are some much more coarse techniques available that we might be able to make use of. And so, for example, people in Oxford, like Charlie Stagg and other people around the world, have shown that when you use TDCS, you can get a reliable and reproducible reduction of GABA underneath the anode. And simultaneously, there's been a great amount of development of techniques to measure the amount of GABA there is in the local network using MR spectroscopy, which is particularly good at 7 Tesla. And so we took this same paradigm back into our 7 Tesla, now with TDCS as well. And what we're trying to show is that this cross-stimulus suppression is there at time point 1. It disappears at time point over the course of hours and days. But then if we stimulate, this suppression will start emerging. And note that would be quite an interesting prediction, because we've reduced the excitability of cortex, but we've measured a suppression that would not otherwise be there, so less activity. Okay, so this is what happens if you do that experiment at 7T in humans. You can replicate first the same experiment. So here's a replication showing that the effect is present at the first time point and goes away overnight. And here's the location of the electrodes. And here's what happens to GABA when you put the TDCS on. You get a reduction in GABA of something like, 8-10%. And then when you reduce GABA in the local network, suddenly you can measure that cross-stimulus suppression again. And what's more, in that same voxel, if you look across subjects, you find that the more you are able to reduce GABA, the more the memory or the association can be expressed as a cross-stimulus suppression in cortex. So we think that suggests that locally in that network, in the LOC, these associations can be stored in balanced excitatory-inhibitory ensembles. Okay, so now I'm going to move on and start talking about ways that you might encode things globally and explicitly. And so to do that, to explain to you what I mean by a global code, I'm going to borrow some ideas from what rodent physiologists do when they have rats running around mazes. This is a rat running on a linear track. And you can see that if he wants to know how to get from one place to another on the linear track, what he has to store is the relationship between the different nodes or place cells on that track. And so what you'd need to store to really understand that track is this matrix here. It's the matrix of transitions, which tells you which one of these blue dots is next to which other one of those blue dots. And so it's a very simple matrix in this particular case because the blue dots are only next to their neighbors, but you can imagine how that would become a much more complicated matrix. But if you then want to figure out what it would be like to move along, to move to the next place in your world, then all you have to do is multiply your current location by this matrix, and you'll end up at this next place. And if you wanted to move to 2, or if, say you wanted to jump to 3 or 4 time points in the future and guess where you're going to be, then you can just multiply yourself by this matrix again and again. But people that are used to dealing with matrices, like I'm sure everybody in this audience is from their general linear models in SPM, will know that all of those matrices, A squared, and A cubed, have the same eigenvectors. Here are these eigenvectors here. And you could encode all of this relational information. Instead of in that matrix, in a local code, you could encode it in those eigenvectors, which would be a global code. And those eigenvectors would have some interesting properties. There wouldn't be one cell that encoded one transition, but instead a multiple of cells would all encode a little bit of each transition. And there are some really important benefits to doing that. I've just shown you that if you do that, you can easily jump 3, 10, as many places ahead as you want to without having to simulate the entire route. Yeah, just to be clear, a local code only knows about neighbours. If you want to get from one place to another, you have to pass through every midpoint. But a global code, every cell knows a bit about the whole structure, and it makes it really easy to jump around this network and to navigate through your knowledge. So I'm just going to have 3 slides of a little bit of maths. Don't worry if you don't follow this. I'll be back to some data soon. This is just showing that the matrix on the left shows that local code again, but shows that it can be represented perfectly by combining the global codes together, V, D, V transpose. Don't worry if you don't understand what that's saying, but do notice that just by changing the way I weight the eigenvectors, using e to the d rather than d, I can do something clever and change this matrix from encoding the local knowledge to encoding the global distances. So this now, this matrix now tells me, if I'm over here, it tells me, oh, look, it's a long way to the far end. And if I'm over here, it tells me, oh, it's a long way to both ends. And so, in fact, this matrix contains all the information about the world. It tells you all the information about how far it is between any points in the world. And you can get there, if you're using a global code, you can get there really simply. The computation is easy. So what you've done here is you've moved a difficult problem, and you've moved it out of the realm of computation and into the realm of representation. You're representing things globally, which makes your computations really easy. So just to show you this working visually, this kind of idea working visually, here's that same matrix again, and now I've changed just 2 elements in this matrix. You won't even be able to see them until I highlight them in the corner. And if you were encoding that matrix locally, you would have no idea that those 2 elements had changed unless you were placed in their neighbours. But if you were encoding it globally, the representation would take care of that. And in fact, that's a really important thing to do, because that has completely changed the structure of the world. So it's made it a circle rather than a line, because it's joined the ends up, which means that all the distances in this entire matrix changes. But because you've represented it globally in this particular example, all of that is automatically taken care of in the computation. And so you can design algorithms that can do clever things. So here's something that Alain Baron and Tim Muller have done. They've made an algorithm which can plan intuitively. It can find the globally optimal routes in mazes without ever searching more than 1 step ahead. It's only searching in those red boxes, but it's immediately able to find this target. And it can do it because the representation it has is a global one rather than a local one. I'm just trying to convince you that it's a useful thing to have. Okay, so I'm just going to state it now in case you haven't followed why. It's really useful to have global explicit codes, because you can easily jump around the map. You can find optimal paths. If you are coding the world as links, not as elements, you can do clever things like form analogies. You can recognize problems with similar structures, even when all the elements of the problem are different. And if you're really clever, you can form coordinate spaces that map lots of your previous experiences, so that you can have intuition based on experiences you've had before. So they're computationally useful things to have. But do they exist in the brain? And so a good candidate start point for one of these for these things are something called grid cells that exist in the entorhinal cortex of rodents. This is the activity of one grid cell, and you can see already that it's a global code, because this cell activates not at one point in space, but instead at many points in space on a very nice hexagonal lattice. And in fact, if you look at the activity of many grid cells, you'll find out that these cells don't all activate at the same hexagonal lattices, but rather they're shifted from each other, and in fact they have different scales as well. They form a basis set of space for the rodent in the same way that the eigenvectors I was showing you before form a basis set for those transition matrices that I was showing you before. And so the question is, can these grid cells form a basis set for other relational information that's not spatial. And this is now the work of Alexandra Constantinescu and Jill O'Reilly. So there's good news for fMRI if you happen to be interested in studying grid cells. And that good news is that these cells all have the same grid angle. So that's the grid angle I'm showing you there. So these hexagonal patterns all line up. Even though they've got different phases, they all line up. And there are other cells that fire faster when you're running along those angles than when you're running across them. So this means that when you that the actual average activity of all the cells in the entorhinal cortex is bigger when you're running along that grid angle than when you're running at one of the intermediary angles. So there's this six-fold symmetry in the total signal that you can record from the entorhinal cortex in a rodent. So this is good news potentially for fMRI because that means there might be a bulk signal from the whole entorhinal cortex that we might be able to measure at the voxel level. That's good. And so noticing this, some of my friends at UCL, Chris Duller and Neil Burgess, decided to go try this in an fMRI scanner. And so they did a virtual navigation task with subjects moving in the scanner in a virtual navigation. And then they decided to look to see if there was a hexagonal symmetry in the fMRI signal that they could measure, such that if you plotted the fMRI signal as a function of running direction, it would go up, down, up, down, up, down 6 times. This is not a prediction that's easy to make if you haven't got a deep understanding of the problem. It's not a prediction that my mum or dad would have made, for example, which is not always true of cognitive studies in fMRI. If you choose to do that, you can indeed find this rather beautiful, precise signal in the entorhinal cortex, as you can see in the bottom left of the screen there, exactly where you'd predict you'd find it in a rodent. But completely strikingly, you also find it in a network of brain regions that they would call a spatial memory network, but we might call a default mode network or evaluation network or a semantic reasoning network. It's a very striking finding because you wonder what grid cells are doing there, but you might think, oh, they've just got something wrong. But this is one of the very unusual cases where fMRI has made a prediction that you couldn't have made with any other technique, and it's turned out to be right. Because there are no grid cells outside the entorhinal cortex in a rodent, but there are in a human. If you go and measure them during virtual navigation in surgery with a wire, you can measure these grid cells, and they're there in the medial frontal cortex, and they're there in the medial parietal cortex, pretty much exactly where Neil and Christian predicted them to be. So what are they doing there? It's unlikely that all of that swathe of cortex is just there for understanding where we are in physical space. And indeed, as I've just said, this network is a network that everybody here is interested in. It seems to be involved in all of our favourite cognitive tasks. So we set about to ask these questions. This question, could we see a similar signal like this in a task that was, on the surface of it, completely different? And so we spend most of our time doing stimulus-reward learning. And so we did one of these tasks. We trained participants to associate these 6 different stimuli, these 6 different black birds, with these 6 different outcomes, reward outcomes, which are different Christmas signals like Christmas trees and Father Christmases. So this is like any other stimulus-reward learning task, except for the fact that the stimuli are not fractals. They're not independent elements. These stimuli are all related to each other, and indeed you can lay them out in a 2-dimensional map as a function of how long their legs are and how long their necks are. And if you give subjects a great deal of training on this, they become very good at navigating this abstract space and finding these rewards. So indeed, there's a direct analogy then between moving in abstract space now and moving in real space, and therefore we should be able to do an experiment which lets us look for these hexagonal signals. So this is what we did. What you can see on the right is what the participants in the scanner saw, and what you can see on the left is what this means in terms of the trajectory. So they would see birds that were moving in bird space, which means they were stretching, they were morphing. And so you could see a bird morphing, and that's like a trajectory of 0 degrees, because only the neck is moving in bird space. But if you'd chosen instead to just move the legs, then that would be like a 90-degree movement in bird space. And of course, we could choose any angle we wanted in bird space and show them these stimuli. They were always being asked which Christmas symbol were they likely to pass through. That was the task the subjects did. And then an interesting thing to note is you could have the same angle in very different parts of bird space. So these last two birds are both 60 degrees, but what's on the screen is very different. So it's a nicely controlled experiment because what's on the screen has no relationship to the prediction you're making about the fMRI signal. And as I've been implying, we're going to go then and plot these different angles around the circle, and we're going to look for an fMRI signal which has 6-fold symmetry, which goes up, down, up, down, up, down as a function of these angles. Okay, so here is the signal that has 6-fold symmetry in our group of subjects. It's again in that default mode network and again in the entorhinal cortex. And you can play more tricks as well. So you can take a region of interest that you're interested in the ventral medial prefrontal cortex, and you can extract that grid angle I was talking about from one session, and you can do a reproducibility analysis. I can just align my next session's data to that grid angle and just plot it. And now this is now a reproducibility analysis showing that there is hexagonal symmetry in that second session as well. And so you can see hexagonal symmetry because it's all of the red bars are aligned to those 6 grid angles, and all of the grey or black bars are misaligned to those signals. So that's kind of cool. That works in the VMPFC. It also works in the entorhinal cortex, and it's only true for 6-fold symmetry. If you'd chosen 7 or 5 or 4 or 8-fold symmetry, which are the other ones around there, you would never have found such an effect. You can then bring your subjects back a week later and ask them to do the same task again, and if you do that, you find that the grid angle, as it does in rodents, is maintained over the course of a week's delay. And again, it's only for the 60-degree ones. And then you can go and look at whether it affects their performance on the behavioural task, and it does. So the more grid-like or hexagonal activity they have, the better they are at being able to remember which Christmas signal is associated with which bird. Okay, so I think, I guess you know what I'm trying to tell you. I'm trying to tell you that these grid cells might be present for non-spatial domains, and when you do an experiment which is very analogous to a spatial experiment conceptually, but very different in terms of the experience of the subjects, they've no experience of, they don't feel like they're moving around space, you can nevertheless see the same signals which may reflect the same neuronal codes. So, but we started off with this kind of problem, which is a problem that looks a bit like a problem of navigating in space, but it's a discrete problem rather than a continuous problem. And you can, and so you wonder if those basis functions are not really necessarily just hexagons, but if they're related to some of those global eigenspectrum-like analyses, then it's possible that they're also interested in these discrete codes with transitions in them. And so we set out with Mona Garvert to look at that kind of problem, to look at that problem. And so to do so, we designed an experiment on one of these discrete grids, where the subjects were doing a task, which was kind of like a cover task, where they were just asked to, they were presented with one of these objects that you can see in front of them, and they were just asked to say, is that thing pointing to the left or the right? Is that ring, that motorbike, that bunny rabbit, is it pointing to the left or the right? But unbeknownst to the subjects, the questions that they were asked were drawn in a very particular way. They were drawn from a random walk along this hexagonal grid. And so if the last question they saw was the ring in the top left of this grid, then they would only ever next have been shown the bunny rabbit, the bike, or the ear. And so somewhere in their brain, it's possible that these relationships have been learnt implicitly, and that may have been learnt with local transitions, or it may have been learnt with a global code in the entorhinal hippocampal system, VMPFC, etc. So the next day they came back and we gave them a subset of these stimuli, and we did another one of these repetition suppression experiments, and we asked, well, if I present the leaf followed by the key, which are neighbours, then maybe there'll be some suppression. If I present the leaf followed by the motorbike, then maybe there'll be a bit of suppression, and if I present the leaf followed by the ring, then there'll be no suppression at all because they're 3 away. And so you wonder so if you can see some kind of distance relationship, linear distance relationship, as you get further away in this map, it suggests there might be some kind of global code underlying the storage of this map in whichever brain region we find. So that's the kind of signal we're going to look for. We're going to look for repetition suppression, which depends on how far away the items are in this map. And so if you do that, again, you find it predominantly in the entorhinal cortex. There are some bits of activity in other parts of this network, but I'm going to focus on the entorhinal cortex. And you see a signal like this, with suppression for distance 1 over distance 2 and distance 2 over distance 3. And notably, that suppression occurs really in exactly the same voxel as you see suppression for spatial distances for subjects wandering around maps of London, or remembering wandering around maps of London. So again, it creates a direct analogy between the types of codes that are used for these non-spatial abstract conceptual tasks and the codes that are used to navigate around space. And so this is the total result of the fMRI signal just plotted there are 7 elements just plotted in a 7x7 matrix, such as, for example, element 2.1 of that matrix would be the fMRI signal that I would record if I presented element 2, which might be the ring, followed by element 1. And so that really is like a distance matrix, because we're expecting repetition suppression. Things that are near to each other should have low suppression, a lot of suppression, low signal, and things that are far away from each other should have not much suppression, so they have big signal. And so that should be like a distance matrix between all of our elements on that grid. And so I should be able to do some tricks like doing multidimensional scaling or PCA of that matrix or something and recover the structure. And so indeed you can do that. If I just do a multidimensional scaling of that matrix, you find that the entire structure that the subjects were measured is encoded directly in the entorhinal cortex fMRI signal. And so, those 1, 2, 3, 4, 5, 6, 7 are different objects, like the ring and the motorbike. And so that structure on the left is the structure that we gave them in implicit learning, and the structure in the middle is what you can recover directly from the fMRI signal. Okay, so lastly, I'm going to just quickly go through a study that I think Erie presented earlier today. I've been talking about neuronal codes, and I think it's a different thing to how most people talk about controlling behavior. Most people are talking about cognitive behavioral models that have things like surprise and prediction errors in them. And so I always thought, when I was working with surprise and prediction errors, that it was very interesting to be able to see those signals, but I always wondered what they did to the representations in the brain. And so what Erie's done is he's done a sort of combined study that combines these representational approaches with a more classical decision-making approach for measuring these prediction errors. And he's trying to ask, how do those prediction errors impact on those representations of the type that I was telling you about before? And so this is what he's done. He's done an experiment where there are these associations between a stimulus, like a bell, and an outcome, like a Starbucks card or an iTunes card. And so that's very similar to the types of experiments I was telling you about before. But the difference is that these ones change over time, and the subjects, much like in many experiments you guys have been, many people have done before, the subjects have to follow these associations in order to work out what the best decisions are to make. And so this is the kind of thing they're asked. They're offered a stimulus, a screen like this. You can have 55p at Starbucks or 45p at iTunes. Do you want the star or the bell? And so they have to remember, oh, the star leads to iTunes, the bell, well, okay, I prefer that. They have to figure out that they think the bell leads to Starbucks, so they'll take that because there's more money associated with that on this trial. And then they might find out that actually that led to iTunes instead, and so they're going to get a big prediction error on this trial. So they're going to get two types of prediction error here. They're going to get a prediction error which says that's different from what I found because I got iTunes instead of Starbucks. But they're also going to get a prediction error which says that's worse than what I found, than what I expected, because they got 45 reward points rather than 55 reward points. That's the kind of task that Erie's done. But critically, he's done that embedded in one of these representational fMRI studies, such that he has a learning block between each. Has a learning block in the middle, but surrounding each learning block, he has one of these cross-stimulus suppression blocks, so he can try to work out what the association is neurally between all of the elements in this experiment. And so if I just look at those cross-stimulus suppression blocks first, you can find out where is that link strength, where is the model that the subjects are currently working with stored in the brain. And again, Erie is able to find that in the parahippocampal, entorhinal, and hippocampal cortex, in the hippocampal formation, much like in the previous studies I showed you. But as I said, there are these two different types of prediction errors that you can find in the other trials, the decision-making trials. So here, Erie's shown that if you get more reward than you expected, you get a reward prediction error, and you can find that in the ventral tegmental area. But if you get a different type of reward than you expect, then you can find signals like that in the orbitofrontal cortex, and that's very much in line with Erie's entire research program. In the particular experiment that Erie's done, what you really should do if you're the optimal subject is ignore that reward prediction error signal completely, because the rewards change on every trial randomly, but use that identity prediction error because there is some structure to which identity, to which stimulus leads to which reward, and that's what you have to learn. And so the question that Erie's then going to ask, very cleverly, is this outcome signal, this prediction error signal that I've measured in many studies before, how did it change that model of the world that is being stored in the hippocampus? So how did it change the suppression between here and here? And so I'm going to look at the difference in suppression between time point t minus 1 and time point t plus 1. That's the change in suppression. That's how much one trial changed my model of the world. And I'm going to model that as a function of how much the intermediary trial, how much prediction error signal there was at the intermediary trial. And you can see that the orbitofrontal signal predicts on a trial-by-trial basis how much change there will be in the hippocampal and parahippocampal suppression. So I think of this as a kind of way of doing functional connectivity analysis with the word function meaning something different. Because here we know what the function of that orbitofrontal prediction error is, and we know what the connection is changing. It's changing the model of the world in the hippocampus, so the orbitofrontal cortex is sending a prediction error to the hippocampus in order to change its model of the world. And that's also true for the VTA reward prediction error, but only in those subjects who learn from those irrelevant rewards. So I've tried to describe you how these models of the world can be encoded in these hippocampal formation signals and in hippocampal formation structures, and I'm now I'm showing that the signal errors, the prediction errors that we're much more familiar with, interact to update and cause learning in those representations of the world and of the world. So that's all I'm going to say. I've got 28 seconds left. That's great. So I've tried to explain to you in this talk that local associations can be stored in balanced ensembles across the cortex, that grid cells conceptually might encode global explicit associations, that these global relational codes can encode non-spatial dimensions as well as spatial dimensions, and perhaps they would also work in worlds that are not even continuous. Thanks very much indeed. I don't know what the formalities are here. Do I take some questions or do I just get down? There was a question. There's not.
I have a question. Do you think there is really any discrete space in semantics?
Do I think there's any discrete space in semantics?
Yeah. And do you think, in other words, in your last example, between the Santa Claus and the duck or whatever it was, in some sense, if you pack discrete associations close enough in a way, then there's...
I think that's true, yes.
And then there's some place where you're approximating a continuous
I completely agree with you. And I think that if you encode things globally, that it becomes a completely a distinction that isn't worth making. I agree with you. Yeah.
This is awesome and inspiring. I was wondering about the higher dimensional spaces, like a three-dimensional space. Is there something like a grid code for that? I mean, in 2D, grids emerge from non-negative PCA, but in the higher dimensions, can we hope for such fMRI signatures?
So it's a very exciting question. It is true that three-dimensional grid cells exist in bats. They are close-packed. So when bats fly around, they are close-packed, like hexagons are close-packed. I think that if you did not have an equal experience of your two dimensions, even in two-dimensional worlds, then you wouldn't have hexagons even in two-dimensional worlds. And so I think that when you go up to really high dimensional spaces, it'll just depend on the covariance between your experiences. And so I think that's one very attractive thing about the PCA way of thinking about grid cells, is it tells you exactly how it will work in high-dimensional spaces, and we're just starting a monkey physiology experiment trying to figure out if that's true or not. So hopefully in 2 years' time, I'll have a cell that tells you the answer to that question.
Hi, over here on the right. Hi.
Hi.
Great talk. So you told us a lot about the encoding part of the sensory inputs and how the encoded structure is being transformed, for instance, by the entorhinal cortex. And at least in sort of a classic Shannon-style information theory, you would have the sender, the channel, and the receiver. So I was wondering, what would be the receiver of these codes of the entorhinal cortex?
I mean, I hope that this is how the frontal cortex talks to the hippocampus. That's what I hope. I don't know if it's true or not. I hope that I mean, I could go into a huge waffle about this. I hope that this is the first stage in transforming information about things in the world and their associations into something that might be useful for action and decision. But I mean, I can only speculate now, and I wouldn't be able to do so coherently in front of thousands of people. I can talk to you about that later if you're interested. But I agree, it's a very exciting question and one that we're interested in. Yeah.
Thank you.
Oh, I was going to actually just make this point. Have I gone now? Can I move on to the next slide? I was going to make this point about Oxford. Just to let you know what it's like being in Oxford and how shit we're all feeling right now. This is what my group looked like 3 and 2 years ago, and it's the same now. We're just, none of us are English. Cool.